In today's lecture, we're going to talk about the foundations of genetics, the historical, um, the discoveries that were made in the late 19th, early 20th century, and the modern understanding of the genetic processes. So the genetics that we're going to talk about for the most part is referred to as Mendelian genetics because the monk Mendel established the framework for this science. He essentially discovered the fundamental principles of heredity without knowing about the concept of genes yet. Although he hypothesized that there are genes that serve as the basic units of heredity. Genes are carried on chromosomes that came later. And we know now that genes can be replicated, they can be mutated, or they can be expressed. We're going to start with the Mendelian genetics, but you will see that not all traits will follow the classic Mendelian pattern of inheritance. Nevertheless, his work, Mendel's work, was essential for full understanding of the genetic concepts later. So early, it was believed that traits are inherited in a blending fashion. So offspring traits are the blend of the parental traits. This concept would explain the phenomenon of the continuous variation. For instance, humans have a continuous variation of height. However, in reality, the continuous traits, such as human height, are continuous because they depend on the expression of multiple genes. Mendel was the first to show that the inheritance is not a blending process. Mendel worked with traits that existed in certain distinct classes. For example, violet flowers versus white flowers. He demonstrated that these traits are inherited in distinct forms. The work of Mendel was forgotten for some time and then was rediscovered in 1900s when chromosomal theory of inheritance was formulated. In his studies, Mendel used a pea plant, Pisum sativum, as the model organism. The advantage of this model was that it is a self-fertilizing plant, and it's a true breeding plant. So what is a true breeding plant? So here you have you see the example of two true breeding plants, plants with violet flowers and plants with white flowers. They're called true breeding because when you cross plants with violet flowers to other plant with violet flowers, the offspring is going to have violet flowers only. Same is true for white flower plants. If you cross white flower plants to other white flower plants, the offspring is going to have only white flowers. Other advantages of this model were that these plants are very easy to grow, they quickly mature, and they produce a lot of offspring. So Mendel manually crossed two true breeding plants with two different traits, for instance, violet and white flowers. The parental generation is often referred to as P0, just P, and when he crossed two plants with two different traits in the parental generation, in the first generation, he only saw plants with violet flowers. That first generation, F1, refers to as first filial, which contains offsprings from the parental plants cross. Then plants from the first generation self-fertilized, so these hybrid plants self-fertilized, producing F2, second filial generation, and in that generation, Mendel recovered the white flower trait along with the violet flowers. Now, Mendel studied several traits, including the height of the plant, the texture of the seeds, the color of the seeds, the color of the flowers we just discussed, the size and the color of the pod, and the position of the flower. These traits, for each characteristic, there were two contrasting traits. For instance, as we just saw, white versus violet flowers. So, as we mentioned, he crossed true breeding plants for white flowers and true breeding plants for violet flowers. This cross produced first filial generation in which all plants had violet flowers, which immediately contradicted the blending theory, since hybrid flowers were not pale violet, but completely violet. The self-fertilization of the plants from F1 produced F2 generation, in which 705 flowers were violet, had violet flowers, and 224 plants had white flowers, roughly 3-to-1 ratio. To control for potential differences between the male and female plants, Mendel carried out reciprocal crosses, when these flower traits were swapped between male and female pairs. In other words, a violet flower could be male or female, and white flower could be male or female. So, overall conclusion that Mendel came to is that one of the traits, in this case white flower trait, disappeared in first filial generation, but reappeared in the second filial generation in a ratio 3-to-1. So he postulated that there are dominant and recessive traits. Dominant traits are inherited unchanged, and they are always expressed in hybrids, e.g. violet flowers. Recessive traits are latent in hybrids, but reappear in the generation F2, in this case, white flowers. So since recessive traits disappear in F1, but reappear in F2, that indicates that traits aren't blended. Essentially, each plant has two copies of each trait from each parent in the form of two alleles. If a trait shows a dominant pattern, in this case violet flowers, this trait can be carried by two dominant copies of the genes, or two dominant alleles, or one dominant and one recessive copy, since dominant copy masks the recessive one. A recessive trait can only be discovered if both copies of the gene, both alleles, are recessive. The ratios 3-to-1 were repeated among different traits, as you can see here, and this ratio was later explained by the probability laws that govern the formation of gametes. So now let's briefly talk about probability. Probability can be empirical or theoretical. Empirical probability is based on observation, such as in Mendel's experiments. Theoretical probability is based on the known outcomes and equal chances for every possibility. In probability, if probability value is 1, it means that something is guaranteed to happen. If probability is 0, it is impossible to happen. So, round seed probability in first filial generation, or violet flower probability in the first filial generation, is 100%, or 1, because it resulted from the cross of round seed parent with a wrinkled seed parent. However, round seed probability in F2 is different. there's 75% probability of the plant to have round seeds and 25% probability for the plant to have wrinkled seeds, i.e. 3-to-1 ratio. Mendel also demonstrated that different traits are inherited independently. Now, knowing this, we can later identify the probability of having a certain combination of traits in the F2 plants. But first, let's talk about the sum and the product rule in the probability. So the product rule postulates that probability of two independent events to occur at the same time, one should multiply their individual probabilities. So for instance, the probability of rolling a die and getting a certain number is 1 out of 6. The probability of flipping the penny and getting heads or tails is 50%. So the probability of rolling a 2 and getting heads is the probability of rolling 2, 1 out of 6, multiplied by the probability of getting heads, 1 out of 2, in other words, 1/12. So this product rule is used for situations where two events must occur together. So for instance, the probability of two dominant traits in the second filial generation is the probability of each 3/4 multiplied, meaning that it's 9 out of 60. The sum rule is used for the situations in which we ask whether one or another outcome would happen. So probability of one head and one tail when we flip the penny is going to be, when we flip two pennies, is going to be 1 out of 2, because the probability of having a head and a tail in certain combinations is 1 out of 4. That probably requires some explanation. So there are, if you have two pennies, you can have two possible combinations. So penny number 1, penny number 2. You can have heads, tails, or you can have tails, heads. The probability of these two must be multiplied, but the probability of these two events must be added. So if we apply this to genetics, the probability of exactly one dominant trait in F2 is, whatever the trait is, 3/4 multiplied by 1/4, because the probability of having one dominant and one recessive, plus the opposite arrangement, 3/4 multiplied by 1/4. If you do the calculations, it's going to be 3/8. So in order to avoid the deviations due to the random chance, like for instance getting 3 or 4 heads in a row, Mendel analyzed a very large number of plants. Now let's talk about the molecular basis for Mendelian genetics. As we discussed before, organisms that are diploid have 2 homologous chromosomes inherited from each parent. So they have 2n chromosomal number. Haploid gametes that have n chromosomes are formed by diploid organisms during meiosis. These haploid gametes unite at fertilization and form a diploid zygote. Gene is a unit of heredity that exists on the chromosomes, and it may have variants, in other words, alleles. Here's an illustration of the height gene alleles, two tall alleles in two homologous chromosomes, and the shape of the seed allele. Now, alleles are essentially the variants of the gene on a specific locus on homologous chromosomes. Not every gene has only two alleles. There are examples of genes that have multiple alleles, and we're going to discuss them. Now, alleles determine the genotype. Phenotype is the observable traits, for instance, yellow pods. Well, genotype, as we mentioned, is the genetic makeup. That genetic makeup includes both alleles that are expressed, dominant, and alleles that are not expressed, recessive. So, for instance, plants in the first filial generation had yellow pods, which was their phenotype, but genotypically they had a potential to produce green pods, which appeared in the second filial. So, depending on the combination of alleles, the organisms can be homozygous when alleles at the locus are identical. For instance, P0 plants, that's a typo in Mendelian studies, or heterozygotes when two different alleles are located at the same locus. So, for instance, in the example that you see at the bottom, the combination of alleles for the height is a homozygous combination. The combination of the alleles for the shape of the seed is heterozygous combination. Now, when you look at the cross here, we can see how this allele combination can produce heterozygous plants in the first generation. So let's say we talk about the color of the seeds. So we have true breeding plants with the yellow seeds and true breeding plants with the green seeds. Each true breeding plant can produce only single type of gametes, either capital Y or lowercase y. When these gametes combine during the fertilization, they produce a plant in which alleles for that particular gene are different on different homologous chromosomes, which means that, so the capital, the uppercase Y, refers to the dominant trait and therefore masks lowercase y, which is a recessive trait. So, despite the potential to produce the recessive trait, despite the fact that the plant carries a recessive allele, the dominant allele masks it, and therefore phenotype is dominant. So that means that dominant alleles are expressed in both homozygous dominant organisms, organisms that have two dominant alleles, and heterozygous organisms. Recessive alleles only going to be expressed in homozygous recessive individuals where both alleles are recessive, since there is no masking from dominant allele there. So in pea plants that Mendel studied, each of the characteristics he looked at had one dominant and one recessive allele, so he pretty much got lucky. And Mendel, of course, didn't call them dominant and recessive. He called them expressed unit factor for dominant and latent unit factor for recessive. Now, when we write the allele inheritance patterns, we use the uppercase letter for the dominant and lowercase letter for recessive. And we use the first letter of the dominant trait as the name for the gene. So, for instance, violet flower is V. We don't use W because it's white flower. So the combination of two uppercase V's denotes homozygous dominant organism, which is violet. The combination of upper and lowercase v denotes heterozygous organism, which is also violet. The combination of two lowercase v's refers to homozygous recessive organism, which is, in case of the color, the flower is white. So there are some traits in humans that are dominant versus recessive. So dominant traits are achondroplasia, it's a brachydactyly, I believe it's short, less number of fingers, Huntington's disease, Marfan syndrome, collagen conditions, and many other human conditions. Recessive traits include albinism, cystic fibrosis, dystrophy, inability to process galactose, inability to process phenylalanine, sickle cell anemia, Tay-Sachs disease, and so on and so forth. To figure out what genotype and phenotype the hybrids are going to have, we can use an approach known as Punnett square. So let's define some concepts here. So first of all, let's define the concept of monohybrid cross. Monohybrid cross is the cross between two true breeding parents that differ in only one characteristic. For instance, here it's the seed color. The offsprings are monohybrids, okay? And in this particular case, the dominant trait is yellow seeds. The recessive trait is green seeds. So in true breeding parents producing yellow seeds, we have two alleles that are both dominant. In true breeding parents producing green seeds, we have two alleles which are both recessive. So how do you approach the Punnett square? You need to list all possible parental alleles along the top of the square and the side of the square. So the alleles that can be produced by true breeding plants are only uppercase Y from the yellow plant and lowercase y from the green plant. Now, alleles that are produced by the F1 generation can be either uppercase Y or lowercase y. So you list them on top and on the side. Then you can combine alleles in each box to see what kind of genotype and therefore phenotype this offspring will have. So in monohybrid cross, as we mentioned before, in the first filial generation, all offspring will carry dominant and recessive alleles, i.e. they are heterozygous. But the color will be yellow because the allele for yellow color is dominant. When these plants are crossed in the second filial generation to produce second filial generation, you realize that there are 4 different types of offspring. Well, strictly speaking, 3. The combination of 2 uppercase y's, the combinations of an upper and lowercase y, and the combination of 2 lowercase y's. So genotypic ratio, as you can see from this illustration, will be 1 to 2 to 1. Phenotypic ratio, on the other hand, will be 3 to 1, because both homozygous dominant and heterozygous will produce yellow seeds, while homozygous recessive will produce green seeds. This explains, from the standpoint of theoretical probability, the empirical results that Mendel obtained for the 7 plant traits that he was observing. Now, what if, as you can imagine, if we are talking about a plant with the recessive phenotype, it means that genotypically this plant is homozygous recessive. However, the plant with the dominant phenotype can be either homozygous dominant or heterozygous. To check which one is that, we can use what's called a test cross. So the organism with the dominant trait is bred with a homozygous recessive organism. So this is the unknown allele. So if we get 4 organisms, or all organisms that are produced, demonstrate dominant phenotype, it means that this allele was uppercase Y, and therefore the parent is homozygous dominant. If we observe the ratio of dominant and recessive phenotypes as 1:1, it means that this allele is lowercase y and the parent was heterozygous. Now, test crosses, while not produced in humans, obviously, but it is used in plant and animal breeding. So this test cross essentially confirms the Mendel's law of segregation or equal separation in alleles. Now, in humans, test cross cannot be performed for a variety of reasons, including ethical and technical reasons. So instead, we use pedigree analysis. We're going to discuss pedigree analysis using examples of autosomal dominant and autosomal recessive traits. So alkaptonuria is an autosomal recessive trait. In other words, the person who has symptoms or affected has to have 2 recessive alleles, has to be homozygous recessive. It affects the metabolism of amino acids tyrosine and phenylalanine. So in this illustration here, the affected individuals are labeled blue. So since this individual is affected, it has to be homozygous recessive, and since this individual is affected, it has to be homozygous recessive as well. You can see that practically this is a test cross. So we can conclude, since the offspring has homozygous recessive phenotype, the only way it can happen if the parent too has heterozygous genotype. This. Now, in order to produce this offspring, the homozygous recessive offspring, when mating with another homozygous recessive offspring, this is another example of essentially test cross. So the genotype of this parent will be heterozygous. Now, if 2 unaffected parents have an affected child, that is possible only if each parent is heterozygous. Now, of course, we can't know whether this offspring is homozygous dominant or heterozygous. Now, in case of autosomal dominant trait inheritance, where red are affected, you can see that even though this parent is homozygous recessive, this parent is either heterozygous or homozygous dominant, and we cannot conclude because there's only one offspring here. Well, actually, we can conclude, because if you look at all the offsprings, so they're affected, not affected, affected, not affected, affected. So if this trait right here is, if this parent is homozygous recessive, then in order to produce both affected and unaffected, in other words, homozygous recessive offspring, this parent has to be heterozygous. So by studying the pattern of inheritance and knowing whether this is an autosomal inheritance, one can conclude about the genotype of an affected individual. Now we're going to talk about some inheritance patterns that do not follow the Mendel's rules. First and foremost, it's incomplete dominance and codominance. Basic rules postulate that there are two alleles, there's no blending, and dominance masked recessive traits. However, there is a situation when the dominance may be incomplete, when heterozygotes show the phenotype that is intermediate between both parents. So for instance, when you cross white flowers and red flowers, you produce heterozygotes that will show the pink appearance. So pink offspring right here. If you then do the self-cross of F1 to produce, F1, just like we discussed before, the self-cross here, the genotypic ratio, as we discussed before, will be 1 to 2 to 1, but the phenotypic ratio will also be 1 to 2 to 1, 1 red, 1 white, and 2 pink. Codominance is the situation when both alleles are equally expressed. So an example would be MN blood group in humans. There are two alleles, M and N, that code for M or N antigens on erythrocytes, red blood cells. Homozygotes express either M or N, but not two of them. But heterozygotes express both M and N antigens equally. So same story, the phenotype and the genotype both are going to follow 1 to 2 to 1 ratio. Another situation that we can observe here is the phenomenon of multiple alleles. So most organisms have just two different alleles, but within the population you can have more than two variants of the same gene. The wild type is often denoted as plus, showing that it is the most common phenotype and genotype, meaning that it is the most dominant one. The variants can be different compared to the wild type. They can be dominant or recessive, and we're going to discuss it using the example of coat color in rabbits. So there are 4 alleles for that trait in the population. These alleles are, these true breeding rabbits produce the brown fur here. The chinchilla alleles produce black tippet white fur, which you can see here. The true breeding Himalayan alleles produce rabbits with white fur and black fur found only on extremities. And true breeding CC gives you rabbits with completely white fur, albino rabbits. So these alleles here are arranged in terms of the dominance hierarchy. This C+ allele is the most dominant over all other alleles. Chinchilla allele is incompletely dominant over Himalayan and albino, and Himalayan is completely dominant over albino. Now, why this happens? So wild-type allele provides the full gene product, mostly allocation of the pigment. Mutant alleles provide less gene product or no gene product. In addition to that, Himalayan phenotype is the temperature sensitive. This allele produces pigment only in the cooler body parts, such as the ear and the nose. This ends our first chapter of the genetics, and we're going to continue our conversation about the inheritance pattern in the chapter 2.